So welcome to our third topic, which is all about hormones, endocrine hormones. Hormones that are endocrine signal at long distance, so they have to be released into the circulation until they find their target receptors on various cells. We'll come back to our old friend, the G protein, in relation to signaling. But before we do that, we need to look at a general outline of the functions of the endocrine system. And alongside of that comes in the master glands. Now, the master glands are basically the hypothalamus and the pituitary, and they work very closely together, joined by a little stalk. And they sort of dominate really in the control of all of the hormone levels and the release of most of the hormones. We'll look at that axis a little bit further in terms of the hypothalamic, anterior pituitary, thyroid axis. Axis in this case just refers to the pathway, the sequence of different hormones stimulating another hormone down that pathway or that axis. We'll then get on to negative feedback because hormones are very powerful things and we want to control them. We have to control the levels. There's various other mechanisms that can amplify them or maybe even downregulate, attenuate them. But in overall regulation, we need to look at negative feedback mechanisms. And we'll do this particularly in relation to negative feedback loops in terms of the thyroid axis, in terms of cortisol secretion. Both of these thyroid hormones, cortisol, are very strong hormones with lots of impacts. So they need closely controlling. Cortisol, again, we'll look at negative feedback loops. And then we'll look at some cellular mechanisms, including the regulation of blood glucose. And this is something we'll come back to when we talk about diabetes and clinical reasoning as well. Thyroid regulation we'll also come back to because in clinical reasoning we'll be talking about hypo and hyperthyroid. So what does the endocrine system do? It's one of the two major systems of the body responsible for communication and coordination. If you think we're talking about hormones, and as I said to you, endocrine means long distance. So these hormones are going all the way around the body in the circulation. So if it's one of the two major systems, what do you think might be the other one? Well, the nervous system obviously is also about communication, but it tends to be much quicker to respond. And it often works alongside the endocrine system in terms of outcomes and in some cases in terms of the release of the hormones. As we said, endocrine hormones travel around the body and they stimulate responses in cells via signaling. Therefore, only cells that have specific receptors will be able to respond. So this hormone, because the obvious thing is it's circulating around, how does it know where to go? Well, it doesn't know where to go. If it finds a receptor to which it can bind, then it will bind. That receptor will be on a specific cell and it will have a specific outcome. I did warn you, specific is my favorite word. So target cell receptors must be specific for the hormone to bind. Any other hormones floating around are just going to do what the little green ball there did, bounce out of the receptor, not bind. These receptors, in this case, in that picture there, we have an extracellular receptor. In other words, it's a plasma membrane protein that's sticking out of the membrane on the surface of the cell. But receptors can also be inside the cell. They can be within the cytosol, even within the organelles, or in the nucleus. And if you remember when we talked about transcription, then we talked about a transcription factor binding to DNA. The other thing, remember, is receptors can be up or down regulated. So we can exocytose some more receptors to the surface of the plasma membrane or within the cell, or we can take some down. We can take some receptors away. And of course, if you remember, what also can change about them when we talked about signaling is their affinity, their attractiveness to their ligand. In this case, the ligands we'll be talking about will be hormones. So, as we said right at the beginning, the master glands really are the hypothalamus and the pituitary. So many of the hormones are controlled by the hypothalamic-pituitary axis or pathway. Now the hypothalamus at the top of this axis forms part of the limbic system. You can see it there in the nice turquoise colour. That's the hypothalamus sitting there, right at the bottom of the limbic system and at the top of the brainstem. There's our hypothalamus. It's joined to the pituitary below it by a little stalk, and that little stalk has a fantastic name. It's called the infundibulum. Just a little stalk. And below that, you can see, we have the pituitary. The pituitary gland is divided into the anterior pituitary and the posterior pituitary. And both of these have slightly different ways of communicating with the hypothalamus. So the pituitary is divided into the anterior pituitary. In old-fashioned texts, you might see this called the adenohypophysis. Hypophysis refers to pituitary, adeno in terms of the anterior part. And the posterior pituitary may be called the neurohypophysis. Again, hypophysis for pituitary, neuro because of the pathway, the axis that we'll talk about in a little while. So first of all, let's talk about the hypothalamic anterior pituitary axis. So this is the pathway between the hypothalamus and the anterior pituitary gland. The hypothalamus synthesizes through protein synthesis and secretes releasing and inhibiting hormones. These hormones then travel to the anterior pituitary to stimulate the release of tropic hormones into the general circulation. You may also see the word trophic with an H in it. Trophic really should relate specifically to hormones that promote growth. Tropic is a general term for the hormones that come from the anterior pituitary and that then act on other glands rather than having a direct action. So the hypothalamus synthesizes, secretes its releasing and inhibiting hormones. These hormones then get down to the anterior pituitary, down that little infundibulum, and there they stimulate the release of tropic hormones. These tropic hormones then go into the general circulation to their target glands or tissues. So the tropic hormones stimulate other glands to release hormones that can then bind to or within target cells. That's the hypothalamic anterior pituitary axis. So a good example might be corticotropin releasing hormone, CRH. This is synthesized and secreted by the hypothalamus. Note, it's a releasing hormone. It travels to the anterior pituitary and it stimulates there the secretion of adrenocorticotropic hormone, ACTH, from specialist glandular cells within the anterior pituitary called corticotrophs. These little trophs or tropes in the anterior pituitary are the epithelial cells that form the anterior pituitary gland. They are all specific and they have slightly different names depending on what they're going to release. ACTH then travels to the adrenal cortex, sat on top of the kidneys, the adrenal glands, the cortex being the outer bit. They travel there via the circulation, and there they will bind and they will stimulate the secretion of cortisol. So this is a classic axis. We've got corticotropin releasing hormone, stimulates the release of adrenocorticotropic hormone, which then stimulates the release of cortisol. So let's look at those hormones. Now, I don't expect you to remember all of these. We are going to go through a few specific ones. Corticotropin releasing hormone at the top there from the hypothalamus, we've just talked about. Where you see the little percentages, so 20% for corticotrophs, that's the percentage of the anterior pituitary that is made up of those specific cells. Thyrotropin releasing hormone, please note that it is not thyroid releasing hormone, it's thyrotropin releasing hormone, TRH. Again, synthesized, secreted by the hypothalamus, travels to the anterior pituitary and stimulates the secretion of thyroid stimulating hormone from thyrotropes. Sorry, these are tropes with no out and H. What if we've got trophs above? There's only 3 to 5% of the anterior pituitary made up of these thyrotropes. Below that, in terms of the hypothalamus, we have growth hormone releasing hormone, GHRH, and we have growth hormone inhibiting hormone. And this will regulate the release of growth hormone from somatotrophs. So obviously, if it's a releasing hormone, it will stimulate the release. If it's an inhibiting hormone, it will inhibit the release. Gonadotropin releasing hormone is all about stimulating the release of follicle stimulating hormone, FSH, and luteinizing hormone from gonadotrophs in the anterior pituitary. Then right at the bottom we have prolactin inhibiting hormone, which incidentally is dopamine, and prolactin releasing hormone. And they regulate the release of prolactin from lactotrophs, also known sometimes as mammotrophs. So you can see we have, in terms of the anterior pituitary axis, we have two sets of hormones. We have one synthesized and secreted by the hypothalamus, and we have the tropic hormones synthesized and secreted by the anterior pituitary. So let's go through that in a little bit more detail. At the top there, you can see the hypothalamus. Ignore the little squiggly image inside it for the minute. That's joined to the anterior pituitary, or the pituitary in general, by the infundibulum. And we're going to be focusing here on the anterior pituitary, labelled there. Now, up in the hypothalamus, we have nuclei. Because this is the central nervous system, wherever we get a collection of the somata, these are the actual bodies of neurons, where we get a collection of those and their synapses, we call those little areas nuclei. And these are neurosecretory cells. So they're like neurons. The only difference is, instead of exocytosing from their axon terminals, neurotransmitters, they're exocytosing hormones. So they work in exactly the same way as normal neurons, but they're called neurosecretory cells because they release hormones. So there in the hypothalamus, you can see the ventral nucleus. The ventral nucleus in the hypothalamus synthesizes, those neurosecretory cells in there synthesize the releasing or inhibiting hormones. So releasing hormones, we're focusing on releasing here, are synthesized in the neurosecretory cells within the ventral nucleus of the hypothalamus. They travel in vesicles. This is just like neurotransmitters. They travel in vesicles down short axons in this case, and those short axons end in axon terminals at the top of the infundibulum. So still within the hypothalamus, albeit within the base of the hypothalamus. When required, they're exocytosed from those axon terminals, and then they diffuse, so they're let loose into the intracellular fluid, they diffuse into capillaries. These capillaries form the primary capillary plexus. This is a specialized little circulation related to the hypothalamic-pituitary axis. So they're exocytosed from the axon terminals, and then they diffuse. Oh, there's a capillary. I'll diffuse into there, get into this nice little blood supply that's been provided for me that forms this primary capillary plexus. Plexus just means network. They then travel down the infundibulum via the hypophyseal vein. The hypophyseal vein takes them down to the secondary capillary plexus, and now we're in the anterior pituitary. So initially they're exocytosed from these neurosecretory cells, and then the releasing hormones travel through a sort of internal circulation within the hypothalamus, the infundibulum, and the anterior pituitary. The releasing hormones, when they get to the anterior pituitary, then diffuse out of that secondary capillary plexus, and they have a little hunt around and they find a specific receptor that they can bind to, and this specific receptor will be on a specific glandular cell in the anterior pituitary, depending on what that hormone is, that releasing hormone. When it binds to its specific receptor on specific glandular cells, it stimulates the release of the associated tropic hormone from those cells. The tropic hormone then travels out of the anterior pituitary and into the general circulation. Eventually, when it finds its specific receptors, it will bind to them wherever that is. So generally in the target gland. This internal circulation, the primary capillary plexus, the hypophyseal portal veins, the secondary capillary plexus, form something called the hypophyseal portal system. And this is the pathway of the releasing hormones in order for them to get to the anterior pituitary to enable the release of tropic hormones. The hypothalamic-posterior pituitary axis is in some ways a bit more straightforward. We're only talking about two possible hormones here. The posterior pituitary receives either ADH, antidiuretic hormone, or oxytocin from the hypothalamus. It then secretes that same pair of hormones directly into the circulation. So it's a little bit more straightforward here. So the neurosecretory cells, in terms of the hypothalamic-posterior pituitary axis, have long axons. Those long axons travel all the way down the infundibulum, and their axon terminals are in the posterior pituitary. So therefore, when they're needed, ADH and oxytocin can be exocytosed from those axon terminals directly into the posterior pituitary. No hypophyseal portal system involved here. From the posterior pituitary, those same hormones that were synthesized in the hypothalamus, oxytocin and or ADH, enter the general circulation and travel to their targets. So you can see here we've only got one set of hormones, if you like. So whereas there are two sets of hormones in the hypothalamic anterior pituitary axis, because we have releasing hormones that stimulate the secretion of tropic hormones, in this case, in the posterior pituitary axis, there is only one set. Those hormones are synthesized by the hypothalamus and they're secreted by the posterior pituitary into the general circulation. And as we said, there are also only two hormones produced by this axis, oxytocin and antidiuretic hormone. So let's have a look at that in our diagram. So you can see at the top there, in this case, whereas we had the ventral nucleus, only one of them, in the hypothalamic anterior pituitary axis, in this case we have two nuclei. We have the supraoptic and we have the paraventricular nuclei. They still contain neurosecretory cells, but look at the axons. They go all the way down the infundibulum with the axon terminals in the posterior pituitary. So ADH and or oxytocin are synthesized by the neurosecretory cells in the supraoptic and the paraventricular nuclei of the hypothalamus. They travel down the infundibulum, again in vesicles within those long axons. They're then exocytosed from the axon terminals when needed within the posterior pituitary. They diffuse into the general circulation and travel off to their target cells. So the hypothalamic-posterior pituitary axis, we only have one set of hormones. So let's talk about the hypothalamic anterior pituitary thyroid axis. So here we're taking it down to the thyroid gland. TRH, thyrotropin releasing hormone, is exocytosed from the axons of the neurosecretory cells. This will be the ventral nucleus at the base of the hypothalamus. So remember those little short axons, exocytosed at the top of the infundibulum. If you remember, it then diffuses into the primary capillary plexus, down the infundibulum via the hypophyseal portal vein, into the secondary capillary plexus, which now puts us in the anterior pituitary, and then it diffuses out to bind to those specific receptors for TRH on the thyrotropes in the anterior pituitary. This stimulates the release of thyroid stimulating hormone from the thyrotropes. TSH travels through the circulation to the thyroid gland. There it binds to G protein coupled receptors on the thyrocytes. And via cyclic AMP, remember cyclic AMP, it stimulates the release of thyroxine, T4, and triiodothyronine, T3, into the circulation. So let's talk about the thyroid gland. The thyroid gland has these pockets or follicles. These are filled with a colloid solution, which is mainly made up of a protein called thyroglobulin. Around the edge of these follicles, we have the thyrocytes. These are cuboidal glandular epithelial cells. The thyroglobulin inside is composed of tyrosine amino acids. And when we talk about the classes of hormones, we'll be talking about thyroid hormones as derivatives of tyrosine, also known as amine hormones. So we have tyrosine amino acids and iodine to combine to produce T3 and T4. T3 has 3 iodines, T4 has 4 iodines. A few T3 only, but up to 30 T4 molecules are stored in each thyroglobulin, and the follicles themselves usually hold sufficient thyroid hormones for up to 2 to 3 months. And this is often why any hypothyroid condition is insidious in onset. It's a slow onset because we have these large stores. So when TSH binds to the thyrocytes, what happens is the thyroglobulin is reabsorbed into the epithelial cells, into the thyrocytes, via pinocytosis, remember that was cell drinking, and then proteases in lysosomes within the thyrocytes cleave the thyroglobulin, and this frees up T3 and T4. T3 and T4 then diffuses out of the thyrocyte via specialist channels and into the circulation. When they get out, T3 and T4, as we said, they are amine hormones, and as we said, T4 has 4 iodine atoms, whereas T3, of course, has 3, hence the numbers. T3 is actually much more active than T4, and often T4 has to be deiodinased, in other words, an iodine taken off, to make it T3 before it becomes active. But we are releasing from the thyroid gland 20 times more T4 than T3. So most of the thyroid hormones released are in the inactive T4 form. So it circulates in the blood, bound to a plasma protein, a thyroxine binding globulin. Globulin just means protein. And it's inactive when it's bound. So this is why there is so much significance placed on the levels of free T3 and T4. Now, technically, they are lipophilic. In other words, technically, they can diffuse through plasma membranes. But iodine carries a negative charge. And if you remember, nothing with a charge can diffuse through the plasma membrane. So this negative charge that the iodine gives to T3 and T4 prevents passive diffusion, non-facilitated diffusion. So it has to be transported into the cell by these iodothyronine transporters. Once in the target cell, T4, remember, is a bit inactive because it's got its 4 iodines, is broken down, catabolized by a deiodinase. This is an enzyme that cleaves off the iodine. It then becomes the more active T3 as and when it's needed within the cell. Thyroid hormones, and in particular, of course, T4 because it's more inactive, are slow acting and they can remain in the circulation or even stored in the target cell for days or weeks before they're activated by the deiodinases. In some cases, there are various deiodinases that can inactivate them within the cell. We'll do more on this in clinical reasoning. So what do? They increase just about everything. They increase cardiac output, increase heart rate, increase respiratory rate, but overall they increase basal metabolic rate. In other words, they increase the speed with which all the processes, the intracellular processes happen, including the synthesis of ATP. They also recruit a couple of mates. So here we have the catecholamines. This is noradrenaline and adrenaline. So this gives us a sympathetic response, in other words, the sympathetic nervous system, which of course is also upregulatory. So this strengthens the effect of the thyroid hormones. Because we've got an increased basal metabolic rate, we've got everything upregulated, we need the increased metabolism of proteins and carbohydrates to provide energy. And because of all this metabolic upregulation and various other mechanisms, body temperature rises. Going back to our hormones. Now, the problem is, as we've seen in the case of thyroid hormones, they can have really significant effects. So the levels have to be controlled very carefully to maintain homeostasis. In other words, to maintain bodily functions within normal ranges, within acceptable ranges. So homeostasis is a healthy, stable state that is maintained by the constant adjustment of biochemical and physiological pathways. Hormonal homeostasis is maintained by negative feedback mechanisms. So to think about a negative feedback mechanism, it's fairly straightforward. Think about your central heating system. You have a thermostat and that monitors the room temperature. When the temperature drops below whatever level you've set it at, my little picture there's got 20.5 or 21. When it drops below the set level, then the thermostat will send, in this case, electrical signals rather than chemical signals to the boiler to turn it on. Boiler turns on, the temperature goes back up. When it reaches the set level, the thermostat then tells the boiler to turn off. So effectively, we are maintaining heat within an acceptable range. Of course, when the temperature drops below the set level again, off we go again. So let's have a look at our thyroid hormones in terms of negative feedback. So at the top, we've got our hypothalamus. Our hypothalamus, as we know, secretes thyrotropin, releasing hormone, TRH, which travels down via the hypophyseal portal system to the anterior pituitary, which is then stimulated to release thyroid stimulating hormone, TSH. TSH goes into the general circulation, whizzes around, and eventually finds itself in the thyroid gland, binding to those thyrocytes. As a result, T4 and T3 are released into the general circulation. When they're released into the general circulation, of course, they upregulate everything, including basal metabolic rate, and all of this produces heat. You'll see why I'm emphasizing the heat in a minute. And here it is. Negative feedback loops, there's 5 of them to do with thyroid hormones to control the release of T3 and T4 ultimately. One of the 2 long loops is the thermal long loop. And this is about thermoreceptors in the hypothalamus monitoring the temperature of the body. So thermoreceptors in the hypothalamus monitor heat. If the heat goes up because of increased BMR and this resulting heat from our thyroid hormones, then it will be registered by those thermoreceptors and the hypothalamus will stop secreting TRH. What's that going to do? Yes, of course, it's going to inhibit then the release of TSH, T4 and T3. So we're stopping the axis, if you like, right at the top with TRH. The hypothalamus also has a lot of chemoreceptors. In this case, we're talking about chemoreceptors within the hypothalamus that bind thyroid hormones T4 and T3. So when there's an awful lot of them floating around in the circulation, they are more likely to bind to these specific chemoreceptors in the hypothalamus. When a lot start to bind, the hypothalamus knows that the levels of those hormones is high. So it again will stop secreting TRH. And those are our 2 long loops, one thermal long loop, one chemical long loop. Both of these long loops will stop the secretion, the release of TRH, and therefore, of course, they stop the release of TSH and the release of T4 and T3. We have one short loop. So this is our 3rd loop. It's a chemical short loop. We don't have any more thermal loops. They're all chemical now. It's called short because it's not the hormone further down the axis. It's TSH in this case. So TSH, that will then bind to specific TSH chemoreceptors within the hypothalamus again. So the hypothalamus is monitoring not only the level of T4 and T3, it's measuring the levels of TSH. If TSH is high, so there's lots of TSH bound to these chemoreceptors, the hypothalamus again will stop secreting TRH with the same ongoing effects of inhibiting the whole rest of the axis. And then we've got some ultra short loops, 2 of these, which completes our 5 loops. The TRH ultra short loop is about specific chemoreceptors, again in the hypothalamus, and these are monitoring the levels of its own hormone. So we have chemoreceptors in the hypothalamus that will bind TRH that is produced by the hypothalamus. It's a bit like an autocrine action in cells. So if TRH levels are high, lots of these chemoreceptors will be bound. And of course, the hypothalamus will stop secreting TRH again. One more loop is our TSH ultra short loop. So this is the first time we've come across the anterior pituitary having chemoreceptors. But it does. It has specific chemoreceptors for TSH, its own hormone. So again, just like in the hypothalamus, if TSH levels are high, it's more likely to bind to these TSH chemoreceptors in the anterior pituitary. And what's the anterior pituitary going to do? Yes, of course, it can't stop TRH. It has no control over TRH. All it can do is stop secreting TSH. Of course, that is still going to have an effect on our thyroid hormones T4 and T3, because if there's no TSH to bind to the thyrocytes, we won't be releasing T4 and T3. So those are our 5 negative feedback loops that involve the thyroid axis. Cortisol only has, well, 3. 2 long loops and 1 ultra short. So, of course, CRH, our cortisol-releasing hormone, corticotropin releasing hormone, stimulates the anterior pituitary to release adrenocorticotropic hormone, ACTH, which stimulates the adrenal cortex to release cortisol. Again, chemoreceptors in the hypothalamus, this time they bind cortisol. If cortisol levels are high, it will be bound to these chemoreceptors, tells the hypothalamus that cortisol levels are too high. Hypothalamus, just like it stopped TRH, can stop CRH. So it inhibits the CRH secretion or release. That, of course, is going to then have an impact on ACTH and cortisol. In addition, we have a loop where cortisol has a negative feedback effect on the anterior pituitary. So here we have the anterior pituitary again having chemoreceptors. In this case, they're chemoreceptors for cortisol. Too high levels of cortisol, too much cortisol bound to the chemoreceptors, the anterior pituitary will stop secreting adrenocorticotropic hormone, ACTH. There's also thought to be an ultra short loop, a bit like our TRH1, where CRH is monitored by chemoreceptors in the hypothalamus, and if CRH is high, the hypothalamus will stop secreting CRH. So those were all very loop-orientated ways of maintaining homeostasis through negative feedback. In this case, we have a different mechanism, really, a different type of mechanism. So if you remember, antidiuretic hormone, ADH, is synthesized in the hypothalamus and it's then released from the posterior pituitary. Now, in the hypothalamus, alongside the thermoreceptors and the chemoreceptors, we have things called osmoreceptors. So these are a bit like little neurons that detect the tonicity in terms of the intracellular and the extracellular compartments. So they shrink by osmosis because they lose water. When the osmolarity, the tonicity in the blood is high, we have hypertonicity. So water is drawn out because of the osmotic gradient. This causes our osmoreceptors to shrink. They then send action potentials along to the relevant hypothalamic neurosecretory cell. In this case, the one synthesizing ADH. So when do you think this situation is going to happen? If we've got a hypertonicity, we've got a lot of osmoles in the blood, in the extracellular compartment compared to inside the in the intracellular compartment. Why would that be? Maybe because we've taken some water out because we're dehydrated. We haven't been drinking enough. So that is going to make the, if you like, the osmotic particles in the blood more concentrated. So it's going to create this osmotic gradient of hypertonicity. So, okay, our osmoreceptors stimulate the release of ADH. This action potential from the osmoreceptor causes the exocytosis through another action potential down those great long axons down, and we get exocytosis of ADH into the posterior pituitary. ADH travels to the renal tubules via the blood, and what it actually does, it increases the aquaporins. Remember, these are the little water channels that we talked about in terms of passive transport. It increases the aquaporins in the renal tubules and collecting duct. Now, most water during glomerular filtration is filtered out into the renal tubules. And that's, of course, then goes on to the bladder, and you know where it goes from there. But we can reabsorb it. And in this case, if you increase the ability to reabsorb the water from the renal tubules, yes, less will go to the bladder, but also you can put water back into the circulation that you would otherwise have lost. And this helps to reduce that osmolarity in the blood because you're diluting it again. So more water is reabsorbed back into the blood, which restores the normal isotonic status. Of course, when this happens, the osmoreceptors reabsorb water, and then they stop sending action potentials to stimulate the secretion of ADH. So this is another non-loopy negative feedback mechanism. Insulin and glucagon counteract each other to maintain blood sugar within homeostatic levels. Glucose enters pancreatic beta cells. These pancreatic beta cells are found in the islets of Langerhans, within, obviously, the pancreas. So glucose enters these. Remember the GLUTs, the transporters we talked about. And we talked about this as a prime example. It's following its chemical gradient because if we've just eaten something with lots of glucose, we will have lots in the blood. But of course the cell doesn't, because the cell keeps using it for cellular respiration. So there will be a chemical gradient. Glucose, once in, is converted by cellular respiration. This will increase the levels of intracellular ATP. Now, beta cells have ATP-sensitive potassium channels that close when ATP binds, and the ATP binds to an intracellular receptor. If we've got an increased level of ATP, then it's more likely to bind to these ATP-sensitive potassium channels. When it does, it causes them to close in beta cells. Because these channels are closed, potassium can't efflux, so it is retained within the cell. We're talking a small amount, a local area of depolarization developing inside the beta cell. This depolarization opens voltage-gated calcium channels. The little voltage sensor reaches threshold, opens the channel. And of course, calcium influxes. Calcium then binds to the cytoskeleton, and that facilitates the exocytosis. In this case, beta cells exocytose insulin. Insulin, of course, lowers blood sugar, and it does it by stimulating GLUT expression in many of the body cells. If you increase the GLUT transporters in cells throughout the body, they are going to then pull the glucose out of the blood and into the cells to be used to make ATP. In addition, insulin also mops up any excess glucose by converting it to glycogen. Glycogen, you stick lots and lots of glucose together in a great long chain, you get glycogen. And that's then stored in the liver. Now, alpha cells are very similar in some respects. So pancreatic alpha cells synthesize, though, and secrete glucagon. So glucagon's role is to increase blood sugar. It's the direct opposite of insulin. Alpha cells still have GLUT transporters in their membrane. They also have ATP-sensitive potassium channels. So glucagon, when it's released, converts the stored glycogen, the insulin stored away, all those little glucose molecules stuck together. Glucagon converts that glycogen back to glucose, and it does this when blood sugar is low. So when we're between meals or whatever, it gives us an alternative supply of glucose if we're not ingesting any. So when glucose is low, because we've used it all up, we increased our GLUTs in lots of cells, they used all the glucose up, we've not eaten since, we've skipped breakfast, whatever. When the blood glucose is low, there will be less entering the alpha cell via GLUT. Remember, these have GLUT too. So there will be less entering. If there's less entering, this is going to have an impact on the availability of ATP within that alpha cell. So there will be less ATP synthesized. The channels, the ATP-sensitive potassium channels, are going to be less likely to bind ATP because there's not as much available within the cell. Now in alpha cells, here's the key difference really. In alpha cells, the ATP-sensitive potassium channels close when there is no ATP bound. So in our beta cell, if you remember, they closed when there is ATP bound. In this case, they close when there's no ATP bound. So they work in the opposite way. And just like in our beta cells though, if we close that potassium channel, we're going to retain potassium. When we retain potassium, it gives us a small area of depolarization. That area of depolarization then causes our calcium channels to reach threshold, calcium channels open, and we have a calcium influx. That calcium influx is going to stimulate exocytosis. But in this place, alpha cells exocytose glucagon, as opposed to beta cells that exocytose insulin. So the alpha cells exocytose glucagon, and glucagon, because it converts glycogen back to glucose, increases blood sugar. And this really is a normal physiological mechanism to maintain blood sugar between meals or between snacks even. So insulin and glucagon, therefore between them and between their exocytosis, dependent upon the availability of glucose, regulate homeostasis of blood glucose. So this is really another feedback mechanism, but in this case we don't have nice long loops and chemoreceptors and all the rest of it. So let's just sum that up. Now, beta cells exocytose insulin. They do this when blood glucose is high, because ATP will also be high intracellularly, and their ATP-sensitive potassium channels will close because it is more likely to be bound by this increase in intracellular ATP. That then causes depolarization, the opening of calcium channels, influx of calcium, interaction with the cytoskeleton, and exocytosis, in this case of insulin. Insulin increases the uptake of glucose from the blood. It sticks loads of GLUTs into the plasma membranes, so all these little cells throughout the body increase their expression of GLUTs. They're more likely to take the glucose out of the blood, which reduces blood sugar. Insulin also, any glucose that's left over in the blood, it will stimulate its storage as glycogen. Both of these effects will reduce blood glucose. Conversely, though, when blood glucose is low, their ATP-sensitive potassium channels will be open, because intracellularly there will be less ATP. Those channels will be less likely to be bound, so they will be open. We have no intracellular depolarization because potassium can freely go in and out quite happily. No calcium influx, no exocytosis of insulin. So when blood glucose is low, we stop exocytosing insulin. But in alpha cells, they exocytose glucagon when the blood glucose is low. And this is because their ATP-sensitive potassium channels close when they're not bound by intracellular ATP. So if blood glucose is low, intracellular ATP will be low, less likely to bind to the channels. But in alpha cells, they close in that situation. When they close, of course, we're going to retain potassium, we're going to have calcium influx, and we're going to have exocytosis. In this case, in alpha cells, we exocytose glucagon. Glucagon goes off to the liver, converts the stored glycogen back into glucose to restore the blood glucose. so it maintains the blood glucose level within homeostasis. In alpha cells, of course, when blood glucose is high, therefore ATP is high within the alpha cell, their ATP sensitive channels will be open. So these potassium channels will open because ATP is bound to them. When they're open, we don't get intracellular depolarization, we don't get calcium influx, and we don't get exocytosis of glucagon. So when blood glucose is high, we don't exocytose glucagon. And between the two of them, they work together to regulate blood glucose. So that brings us to the end of part 1 of endocrine. As usual, try and get this all under your belt before moving on to part 2. So your own notes, your glossaries, all of that. Get that organised and try and learn the bulk of it before you move on to part 2. In part 2, we'll start to look at the classifications of hormones and also the signalling mechanisms of hormones. So before starting part 2, you might want to look back at G proteins, for example. I'll see you in part 2.